they used to load the drugs along with those nanoparticles, and then they'll try to drag those drugs embedded, I mean, nanoparticles into that cancer tumor surrounded area, and then they'll apply some of the external stimulus, like maybe a magnetic field or some sort of like uh, laser light irradiation, etc. Once they do that, what will happen that photothermal efficiency will be enhanced, maybe of temperature rises between 40 to 42 5 degrees Celsius. So that is enough to kill those cancer cells either completely, or at least they can stop the tumor spreading, etc. Okay. And then yeah, basically it helps mainly for the stage 1 and stage 2, as compared to the other traditional drugs like other drugs. They, I mean, although they also does a job like killing the cancer cell or reducing the tumor size, etc. On top of that, they also affect other tissues and organs. So that's a little, I mean, unusual way, as well as since this has a high photothermal efficiency, it can irradiate completely from the human body. Okay, the cancer tumors. So, so I'll introduce you to, I'll show you some of the papers where they have worked recently. So, I mean, various materials has this photothermal efficiency properties. Okay. So one of the materials called MXN, people used to call like 2D titanium based carbide, or I mean, kind of like this. I mean, metals can be replaced with neobium, etc. So neobium, I mean, it has better photothermal efficiency. Okay. So it's also kind of graphene structure, like 2D layered gallery based structures. So you can do all those modification. Maybe like you can dope any kind of like manganese oxide or iron oxide, etc. So that you can control. I mean those. I mean properties with the help of external similar like a magnetic field, etc. I mean, on top of that, I mean, you can also play around with that other irradiation, laser light irradiations. Okay? So this paper, like they have synthesized this titanium carbide using top-down approach, and then they have doped the manganese oxide into the titanium carbide, so that you can play around with the magnetic field also, like I mean, by increasing the magnetic field, how the photothermal efficiency is working and how it is scaling, etc. And then I mean that one they have, I mean, the manganese doped titanium carbide mexin based nanosheet was modified by using polymer based I mean molecules. Okay, so that it can enhance the interactions with the tissues, or the cancer tumor, etc. And then once it is done, and they have introduced this I mean nanosheet into that I mean in vivo cancer therapy using a rat model. And then the total that portion was, I mean, irradiated with the help of laser light, okay, NIR1 region. And then they have seen how the temperature changes, everything, like with help of I mean sensors. Okay. So, so these are like materials part, like top-down approach. You have a bulk sample, bulk ceramics, and then you do different kind of etching so that you can brought down the size into the nano level. Okay, and then they all have done the different process, just like how you do the Hammers process for graphene. And then yeah, you will end up with this kind of surface functionality, which has alcohol functionalities. Okay, that one again, that one used for I mean doping the manganese oxide. So once you have done the doping again, they have used some sort of like polymer based I mean aliphatic chains, so that they can I mean you can change the hydrophilic surface into the hydrophobic, and hydrophobic will better interact with the protein and other part of your bodies. Okay. And yeah, this is some of the pictures. You can see the layered structure of the MX nanosheet, and once it is converted into the nanosheet, you can see the I mean the dispersion of those individual sheets. Okay. Again, the same probably this one is again this was TEM, and this is surface I mean morphology using HCM. This was again confirming the doping, I mean, ions like magnesium's presence in the nanosheets. and where I want to show you. Yeah, here. Yes. Yeah. So look at here. They have dispersed the uh, prepared maxi nanosheet in water at different concentration. Okay, this one pure water and this one has different concentration of MXC nanosheet. And then they have introduced the external field like I mean electrical field, magnetic field, etc. And then they have seen how the temperature changes over the period of time. Okay, so you can see here, okay, in the pure water, I mean there's no changes in the temperature, but the moment when you introduce those electrical field into that highly concentrated or I mean maximum incorporated water, you can see with the time like how the temperature is rising, like from 30 to literally it has reached around 45. Okay, and this is again switching on and switching off, like you I mean do that simulation like with the specific time interval, like switch it on and then how the temperature is rising, and switch it off and then how the temperature is getting reduced, etc. So all those things. And yeah, and then they have introduced that for imaging as well, since you have that magnetic sensing characteristic, like by introducing of magnesium oxide. So you can use them for, I mean, tumor sensing as well, where exactly the tumor is there. That is also can be doable. Okay, you can use them for imaging, and then they are used for how good the tumor eradication efficiency. Okay, so the tumor was induced in the rat model, and then they have given those, I mean, MXC nanosheet into that rat, and then they had triggered those sheet towards to that um, tumor where the, I mean, where they have induced the tumor in the rat. Okay, and then they have irradiated those NIR light to that specific tumor area. I mean, and then they have seen like with the different sample how the tumor size is getting reduced with the help of, I mean, NIR irradiation. Okay, you can see the temperature rises from, I mean 30 to around 55 also. Okay, for the I mean sample which has I mean MXin as well as manganese oxide doped one. And yeah, even any temperature like above 40 45 is excellent for killing those cancer tumors. So one advantage is that, I mean, since we are simulating this temperature with the help of external field like laser light, so the specific that area only the heat will be, I mean, generated. So as a result, the other part of the tissues are the organs that is not going to be affected at all. And then this was a tumor size, I mean, how it was brought down, okay, as well as the volume. And yeah, this is kind of other analysis, like whether the other part of the organs are getting any infection or kind of damages which has happened due to that. So they used to check like before and after how, I mean, the tissue is present. So you can see here, this was the pure controlled one, I mean, which was I mean, cultured from the, I mean, processed from the rat. And then you can see the morphology literally, I mean, there's no change in the morphology. You can see all those connective tissues and then ECM matrix, everything. Okay. So, so you can, I'll upload this paper, you can read, and then you see like this paper was worked on titanium based MXN. And this one they worked with the niobium based, I mean, the same group, research group. They use some niobium carbide based MXN. Okay, you can see how good the tumor size, etc. Like, I mean, it is, I mean, complete eradication of those tumor in, I mean, how much time it took, all those things you can compare it. I mean, literally, niobium has better photothermal efficiency. So as a result, like they can, I mean, get away, I mean, eradicate the cancer tumor completely. Okay, in a very fast manner. So this also like they have done some sort of. So they have done some simulations and then how those gallery space, I mean, can be enhanced and what is the theoretical, reason all those things can be, editorial. So as well as how to theoretical calculation of those photothermal efficiency, all those things you can read here. Well, one difference like the good result, like what they found is that. Yes, I wanted to show you one result was significantly different from the others. So anyway, I forgot. I mean, which result? Maybe I have to look more closely. So that's all about those hyperthermia concept using those. Photothermic efficiency-based materials. So, apart from that, that magnetic characteristics can also be used for targeted drug delivery applications, like with the help of, surfactant or kind of triblock copolymer, like pluronic or any other acrylic-based copolymer. Okay. So pluronic has three different segments, like a hydrophilic and hydrophobic and heterophilic. Okay? Polyethylene glycol and then polypropylene and then polyethylene glycol-based segments. So, so this was the segment, okay, three different segments, okay. That you're gonna, I'm going to chemically linked with those prepared, either micellar or self-assembled structures, okay. So that once you do that, so you'll have a more, free volume or kind of randomness in your self-assembled structures. So you can see here. So once you do that, what will happen? These tracks can be embedded into that. Spaces between those self-assembled structures. Okay, and of course they can embed easily, and releasing is also pretty easy. Okay, so once you do the kind of simulation like temperature changes or pH changes, the drugs can comes away, immediately, as controlled way. As also you can also play around with this magnetic field as well, since the micelle is formed with the help of magnetic nanoparticles. You can do that as well. So while we're using magnetic nanoparticle, of course, they are biocompatible as well as like biodegradability. Certain concentration to certain concentration. And the oleic acid also widely used to stabilize those iron nanoparticle, because iron nanoparticle has got excellent magnetic characteristic. As a result, they get aggregate very soon. Irrespective of the suspension form what you are using. So the oleic acid they used, to tag on the surface of the magnetic nanoparticle. Okay. On top of that, okay, you have those, either alcohol or acid functionalities on the, oleic acid functional, degraded iron nanoparticles. So again, with the help of those either, condensation or the rearrangement reactions, you can tag this pluronic into that surface of your, oleic acid main chain. Yeah, they are chemically linked, okay, covalently linked, and then yeah, then you can do any kind of drug, like you can load the drugs, like kind of anti-cancer drug or any other kind of drug, physically as well as chemically as well. And the advantage is that since you have the magnetic properties as well as those kind of like well designed structure like a pluronic attached coated one. So you can play around with this, I mean, temperature, pH, as well as the magnetic, I mean, field actually. As a result, you can release the drug very slowly or the controlled way as compared to that of other, I mean, modes of release. Okay. And they do have some disadvantage like you cannot take like iron nanoparticle like how much over the concentration you want. You can't do that actually. So iron nanoparticle, of course, they are biocompatible and biodegradable, but up to till certain concentration. So that is also important. And this kind of chemically complex steps like the mole ratio, how much you are taking, all those things are very important. So because pluronic is also water soluble, okay, and the oleic acid is also water soluble. So the at different solvent and they react different, I mean, manner. And the type of, I mean, temperature and the pH, how the reaction you are carrying out to make sure that the oleic acid is getting reacted with the surface of your iron nanoparticle. All these things are very key role, okay, and yeah, this is how it works. Like the drug solutions, and then yeah, and then they are evaporating those ethanol, and then they are adding those another like pluronic based one to make sure that it is getting again, I mean, covalently linked on the chain of your oleic acid, and then you can add the drugs, etc. I mean, later on also once the drug is added, then with the help of those magnetic separation, you can separate those only the drug loaded, I mean, self-assembled structures, and then yeah, wherever you want to release, you can release it. Okay, so you can see here like a doxorubicin based release, which was, I mean, studied using iron nanoparticle based self-assembled structures. So the drug release even till I mean, it can be released till the 30-day or even after that as well. As well. So literally like first initial days, like till fifth days, you can see only amount of like a 30 to 40% of drugs was released, and rest for 60% was released very slowly. Okay. So as I told you, I mean earlier also, like the drug releasing manner is very important. It's not that the initial days itself the drugs get released like 60-80%, and then later stage if they are releasing like 10%, etc., that is not going to help you at all. Okay. Rather, yeah, they helps, but on top of that, they also, I mean, do some side effect with other kind of tissues. Okay. And this is again like how much drug uptake, I mean, with the cancer cells, etc. Like you can see here like few dogs like 2 hours how the effect, and then 24 hours how the effect, and then 48 hours. So literally the cells are getting died, and with the help of nanoparticles you can see like I mean almost I mean all the cells are getting died in 48 hours. Of course the I mean we have to watch out this microscopy, I mean the cell culture things for quite long time. And you can also use for I mean magnetic nanoparticles for MRI scan for the brain stroke, etc. Okay, so I stop here. mm, from next class we'll move on to your metal based advanced engineering materials and their processing as well as the applications. Tomorrow also can we meet in the morning time sometime?
Morning is
Is there
free
any?
tomorrow.
No, if at all, if somebody is canceling, please let me know, like Pritham or Arya.